Thank you for attending the Applied Plant Sciences Seminar Series. Today, I would like to remind everyone that questions will be answered at the end of the talk. You can type your questions into the Q&A box throughout the presentation, or you can raise your hands at the end and we will turn on the mic for you and you can ask your questions live. So I would like to introduce Hannah. Hannah is a PhD student with Dr. Jim Anderson in the Department of Agronomy and Plant Genetics, and her thesis research is on quantifying breeding progress in intermediate wheatgrass, also known as Kernza. She is passionate about feeding the world using sustainable agricultural practices and advanced breeding technologies. She's also an advocate of science communication. She likes to communicate science to learners of all backgrounds and educating future scientists. Hannah is originally from Central Illinois, and she obtained her bachelor's degree in crop science at University of Illinois at Urbana-Champaign in 2017. She then moved to Nebraska, where she studied hybrid wheat breeding and genetics with Dr. Steve Benziger, and obtained her MS at University of Nebraska-Lincoln in 2019. In her free time, Hannah enjoys biking around the cities with her husband, trying new coffee shops, breweries, and wineries with friends, and this is what I like the most. She's spending way too much money landscaping her rental house. And with that, I will give the floor to Hannah.
Thanks so much, MJ, for that great introduction. And thank you everyone for being here today for me to tell you about, share with you my research that I'm planning on doing for my PhD, quantifying breeding progress for domestication traits in intermediate wheatgrass, or Kernza, at the University of Minnesota. So I'll just start out with an outline so you all know where we're going. I'll start with an introduction to intermediate wheatgrass. Then I'll talk about assessing breeding progress over 4 cycles of recurrent selection. Then I'll talk about a GWAS and comparative genomics to identify regions of the genome that are associated with important domestication genes. Then I'll kind of change gears and talk about preharvest sprouting, an important agronomic trait in intermediate wheatgrass. And I'll finish off with developing Kernza education and outreach materials. And while the last portion may or may not be included in my final thesis research, I still think it's important to share because it's vital to the success of this new crop as a whole. So I'll start off with an introduction to intermediate wheatgrass. Intermediate wheatgrass is a dual-use perennial forage and grain crop. So you can get up to 3 harvests on this perennial crop. The grain is marketed as Kernza. So throughout this talk, you might hear me use the term intermediate wheatgrass or Kernza. Kernza is the name that's trademarked by the Land Institute. It's technically the designation for the grain, but just know that I might use these 2 terms interchangeably. Intermediate wheatgrass is the name of the plant, Kernza, the grain. And this grain is used in a number of food and beverages. So if you want to order some Kernza flour for using at home, you can go to the Perennial pantry and order some Kernza flour. Notably, the quality of this flour is different from bread wheat. It doesn't have the same gluten content. So it's more so used in crackers, baked goods that don't need to rise because it doesn't have that gluten strength. It's also used in some beers. So Bang Brewing in the Twin Cities, if you're interested in visiting them and grabbing a beer, occasionally they have a Kernza beer on tap. So keep an eye out for that too. It's Thinopyrum intermedium. It's a perennial cool season grass, which just means that it needs 6 weeks below 40 degrees Fahrenheit to produce grain. It's a near obligate outcrosser, doesn't self-pollinate, and it provides numerous ecosystem services that I'll talk about later. Let's talk about intermediate wheatgrass, our timeline that we're looking at. So in 1945, that's whenever the NRCS first released their first variety of intermediate wheatgrass called greener. And this was a forage variety, meaning we weren't harvesting the grain, we just was using it as a forage. Then moving forward to 1983, that's whenever the Rodale Institute took it on, and they decided that they were going to try selecting for grain quality traits in intermediate wheatgrass. They completed 2 cycles of selection for grain quality traits, And in 2002, the Land Institute took it on. They took 1,000 genets. So I'm going to use the term genet periodically throughout this talk. So I think it's important to define what that actually means, how we use it. A genet is just how we refer to a single genotype or plant or a genetically unique individual. I'll probably use the terms parent and genet interchangeably in this talk. Just know that they mean the same thing, and it's just like a single genotype or plant. And then in 2011, the University of Minnesota started breeding for grain quality traits. We've completed 4 cycles of recurrent selection. We're on our 5th. And in 2019, the very first Kernza variety was released, and then Clearwater. So our first variety release was just in 2019 with this new perennial grain crop. But in the context of other domesticated crops that some of you are familiar with, if you compare intermediate wheatgrass to hexaploid wheat was domesticated in the Fertile Crescent around 10,000 years ago. Same with corn, potatoes, different areas of the world, but all domesticated thousands of years ago. We're working with a very different time scale with intermediate wheatgrass. So 10,000 years ago was hexaploid wheat. In 1800s, that's whenever the earliest wheat crosses were made by humans. The Green Revolution was in the 1960s. And then 1983, that's whenever we first started domesticating Kernza. So very different time scale. However, it's important to note that we have a lot more tools at our disposal now than we did 10,000 years ago whenever crops were first getting domesticated. I'll talk about some of those tools later in this talk. What are domestication traits and what even is domestication? Plant domestication is basically just the action of selecting for traits in a wild species that suit the needs of a human. So in plants, these traits are typically things that make it easier for humans to harvest and eat the grain. In the case of small grains, having less shattering, less grain ending up on the ground, grain that dehisces from the hull for easier milling, and the obvious one, which is better yields. I'll start by talking about plant type. We want a plant that doesn't fall over on the ground, right? We don't want lodging, so a plant that's shorter in stature but still yields well is ideal. Next, spikelet density. So looking at figure D here, we see intermediate wheatgrass on the left compared to wheat on the right. You can see that intermediate wheatgrass has florets that are a lot more spread out along the head, whereas wheat is a lot more compact. So selecting for things like more dense spikelets that are make for a more compact head that's less likely to get broken off and for you to lose half of your yield is ideal. Also, number of seeds, more seeds per floret and more florets per head in a smaller area would be great. Seed size is also an important domestication trait. This figure here compares forage varieties to our current intermediate wheatgrass Kernza varieties. You can see seed size has been improved quite a bit, but still in comparison to wheat, or down here comparing it to wheat is on the left, intermediate wheatgrass on the right. We have a lot of improvements to be made for seed size, which leads to yield also. Free threshing is another important trait. You might hear me use the terms free threshing, or I might call it threshability, its ability to be free threshed. Whenever you look at the top, these are hulled seeds, and bottom are de-hulled seeds. Hulled seeds are just another step that millers would have to go through to get the grain to a state where they could mill it down for flour. So it's not ideal to have hulled seeds because it adds another step. And also whenever you go through the de-hulling process, you tend to lose a little bit of yield, which for obvious reasons is also not ideal. And the last trait, one that I'll also talk about more later, is shattering. We don't want our grain to end up on the ground. We want it to end up in our hand. So breeding for reduced shattering is also really important. So we're going to start off with a poll. Go ahead and activate it. For those who haven't joined, you can still join. I'm curious to hear from the crowd. What benefits do you think a perennial cropping system offers? So this will show up as a word cloud, notably one word at a time. It only takes one word at a time and you can only submit one response. So if you have a multiple word phrase, yeah, include it as one word or put an underscore or something like that. Sequestering carbon, great. I'll probably give another 30 or so seconds, unless we're filling up. All right, reduced tillage, herbicides, less erosion, pesticides. This is fun. Reduce erosion, I'm seeing less tillage. I'm seeing reduce coming up frequently. Awesome, thanks for putting in the chat too, water quality. Yeah, that's great, feel free to do that. Increase soil organic matter, nutrient capture. Yeah, these are great. Sounds like we have a lot of experts in the audience too. All right, we'll give it a couple more seconds. Increase soil organic matter, establish once, reduce tillage, reduce erosion, sequester carbon. Yeah, these are great. All right, thanks everyone. I'll move on. Yeah, why perennials? I almost don't even need to present this slide because I think everyone provided a lot of feedback as to why perennial crops like intermediate wheatgrass would be great. But I'll spend a little bit of time to go over those. So we have long roots that are present year-round in the soil. Looking at this picture here, we compare intermediate wheatgrass to annual wheat. And as you can see, there's a lot more presence of roots in the soil, and throughout the whole year, there's roots present in the soil. So this can lead to potential carbon sequestration and improvement of water quality. So reducing nutrient leaching, which for rural areas can be really important, and soil conservation, reduced erosion if you have roots holding the soil in place year round. In addition to environmental benefits, there's also some potential economic benefits to the farmer. So it's a dual use crop, which means you can have both a grain harvest and a forage harvest with this crop for multiple years too, and reduced inputs. So you're only planting this one time and you'll get to harvest it for 3 years. So a number of economic benefits or potential benefits for the farmer growing this crop. So if it's so great, then why aren't we growing? Then why isn't everyone doing this, right? Domesticating a perennial crop obviously has a lot of challenges, and a major challenge that we see with intermediate wheatgrass is that yields progressively decline each year. This figure here shows some yields of some of our Kernza genets, Minnesota Clearwater here. Remember, that's our first release variety compared to some of the Land Institute's materials. And whenever you look at year 1, year 2, and year 3 yields, you can see they progressively decline, especially in year 3. So a perennial plant wants to put all of its resources into its roots. That's how it survives, right? Which roots are great. We need roots to hold the soil in place. However, we're kind of asking the crop to reallocate its photosynthate from the roots to the grain. So that presents a really unique challenge with domesticating a perennial grain crop, the first of its kind. Moreover, grain quality, which I mentioned a little bit at the beginning of this talk, but Kernza has higher protein and fiber content than wheat, but it also has lower starch and glutenins. And the gluten quality, there still is gluten in intermediate wheatgrass, but the gluten quality is not great for making a loaf of bread rise really nicely. So that just brings attention to the importance of incorporating food scientists into this grand domestication challenge of this perennial grain crop. I wanted to give a brief overview also of how we breed for intermediate wheatgrass at the University of Minnesota. And for those who are not breeders, I urge you to pay attention because it's really not that complicated. And hopefully this illustration will help prove how uncomplicated it really is. So at the University of Minnesota, we use what's called recurrent selection paired with genomic selection. And all recurrent selection is the cyclical improvement of a population or the generational improvement of the mean. So basically you're selecting the best of the best from each cycle, and you're using that to perpetuate the next cycle. So looking at this here, we have cycle 1. Let's pretend that the x-axis is yield for a trait that you're interested in, right? We want higher yields. So we're going to select the best individuals from cycle 1 to intercross and perpetuate the next cycle. So we select those. Oh, look at that, it's magic. We see an improvement of the mean for yield because we selected the best from the cycle before. And then we go to cycle 3 after selecting the best of the best. And again, we see an improvement of the mean for these traits that we're selecting for. So population improvement. That's at the simplest level, that's what we're doing in our breeding program to improve this crop for traits that we're interested in. Another good illustration to combine this genomic selection that I was talking about with recurrent selection. This figure is from Praveen Bashgain, who's actually doing a lot of the breeding work here at the University of Minnesota. But genomic selection is just using genotypic and phenotypic data to develop models for predicting performance based on genetic information. So let's start right here at the training population. Remember, keep that cyclical population improvement theme in mind as I'm explaining this. So we start with our training population, which in our breeding program is around 600 plants or 600 individuals. Our training population is both genotyped and phenotyped. So use that information to then build genomic selection models. However, we also have our breeding population. And our breeding population, we don't phenotype, we only genotype it. Phenotyping is really expensive, really time consuming. So if we don't have to phenotype everything, that's where genomic selection really comes in and helps us out. So we have our breeding population. We don't phenotype everything, but we do genotype it. And we use those genomic selection models that were trained with our training population that was both genotyped and phenotyped to get predicted performances for all the individuals in our breeding population. From there, then we select the best 70 to 100 parents, or best plants as parents. Remember those curves that I just showed you, we're selecting the best of the best to perpetuate the next cycle. Those are shot back into the training population, and then everything, those selected parents are then piped over to our cultivar development part of the breeding process. So cultivar development first starts by intercrossing those selected improved parents for the next cycle. So in our breeding program, we have around 70 to 100 of those best parents that were selected using recurrent selection with genomic selection. They're intercrossed. This figure here shows a crossing block, which usually we have around 10 genets or 10 parents per crossing block, replicated a few times. They're just allowed to open pollinate. Remember, it's an obligate outcrosser. They're allowed to open pollinate, and they're separated from other crossing blocks with quite a bit of space in between, so there's no pollen travel between crossing blocks. The seeds then bulked from those synthetic crossing blocks and put into yield trials. So we need to see how these things actually yield in addition to the other traits that we're interested in. That goes on for a couple years until we get to seed increase, and not very many are selected for seed increase. It's kind of only the best of the best are selected for production. And finally, after seed increase and continued evaluation, we get to variety release, which the very first Kernza variety was MN Clearwater, which was Minnesota 1504 SIN4 seed. It's not that complicated, right? And just to illustrate the power of recurrent selection with genomic selection, I have a couple figures here that I made with some of our early data with shattering and threshability. We've seen rapid improvements for shattering and threshability using this method. So notably on the top left, we have resistance to shattering at breeding cycles 2 and 3. The data just was not collected for cycle 1, that's why it's missing. A lower score for resistance to shattering is better. So you can see even between cycle 2 and cycle 3, we've seen improvements. Moreover, for threshability, comparing cycle 1, 2, and 3, notably cycle 1, they used a slightly different rating system, so they didn't include zeros. If they did, we probably would see more of a linear increase between 1, 2, and 3. But nonetheless, you can see an improvement from cycle 2 to cycle 3 using that same rating system. So recurrent selection works. We've seen the progress. And we want to be able to quantify it a little bit better, which brings importance to what I'm planning on doing for my PhD research. People are really excited about Kernza. Can this breakfast cereal help save the planet? General Mills backs development of Kernza. a cereal that's deeply rooted for good, and the climate-friendly grain Kernza finally hit the big time. USDA funds project encouraging farmers to grow Kernza. And my personal favorite, after a long day of fighting climate change, this grain is ready for a beer. So I'm going to wake you all up with another poll question. I'm curious to see how many acres of Kernza you currently think are planted in the United States. Is it less than 1,000? Yep, 1,000 to 5,000. All right, I'll give you probably 30 seconds to select your answer. Looks like we have a winner. B might be the winner. Those who put B were correct. There's currently 3,500 acres of Kernza in production in the United States as of spring 2021. There's 913 million acres of farmland in the US. So put in perspective, that's 0.0000038% of land area currently planted to Kernza. Or if you've ever been to LAX airport, it's about the same size as LAX airport. Nonetheless, Kernza production is being targeted to areas that are at greatest risk and have the most to gain from having Kernza produced in their area. So this map that I have here shows economic and environment clusters of opportunity. And the early commercialization efforts of Kernza are trying to target its production to these areas that are best suited and have the most to gain from having Kernza grown in that area, which if you're looking at the state of Minnesota, the bottom half of the state has been a target for early adoption of Kernza. So despite its low acreage, it's still being targeted to areas where it can have the most impact. So there aren't very many acres currently being grown of Kernza, and that's because there's a lot that has to happen together in order to actually commercialize a new perennial crop. This figure that I have here is from the USDA NEPA Kernza CAP objectives. It's one of their main graphics for that grant, but I thought it was a really great illustration of everything that has to be working together to get to that end goal of intentional integration and commercialization of a perennial crop. So the first thing is to advance germplasm and trait evaluation. And there's several leads that I'll have incorporated here. Notably, these three individuals who are listed are only the co-leads. There's a whole team in addition to these co-leads that's working on this part of advancing germplasm and trait evaluation. You also need to enhance agronomic and on-farm knowledge. So early adopters or farmers have a lot of really great knowledge, and making sure that that's being shared with researchers and vice versa to improve the agronomics of this new crop. Obviously, improving environmental quality was another thing. You also need to engage education, extension, and policy. So people need to be educated on what this crop even is. Extension educators also need to be educated so that they can relay the message to growers. Policy changes too are important. And lastly, whenever farmers grow this, they need to have a place to actually sell the grain, and end users need to know that they have a reliable supply of the grain coming to them. So developing supply chains and other economic drivers for the new crop is also important. And all of these things built together lead to their intentional integration. If you remember one thing, commercializing a new crop is a team effort, and there's a lot of people involved with this crop. And what I'm planning on doing is only a really small portion of this, but I'm really glad to be a part of this team. Today, what I will be mainly focusing on is advancing germplasm and trait evaluation for Kernza. And at the end, I'll talk a little bit about this education and extension portion, why I'm interested in it. So thanks everyone for sitting through my introduction. Now I'm going to dive a little bit more into what I'm actually planning on doing as part of my PhD. So I'll start off talking about assessing breeding progress over 4 cycles of recurrent selection. I'll talk about my experimental design, how I plan on phenotyping, and lastly about breeding progress and genetic gain. So the Intermediate Wheatgrass parent trial is my main trial for most of my objectives for my PhD. The parent trial is several cycles of our materials all planted in the same experiment. So we have cycle 2, 3, 4, and 5 parents all planted in the same experiment. We just don't have cycle 1 parents because they're still at the Land Institute, and we actually don't know the genetic identity of the pollen source for cycle 1. So that's why we're only looking at cycle 2, 3, 4, and 5 parents. We've seen improvements for many of these domestication and agronomic traits, as I showed in my earlier figure showing cycles 1, 2, and 3. However, we've never actually grown each of these parents in the same experiment. We're always comparing across years, across cycles. So having all of them grown in the same experiment, same environment, allows us to have better estimates of genotypic effects, G by E, things like that. So we're getting better estimates by having everything in the same experiment. My experimental design is an augmented trial design. An augmented design basically just means that I'm not replicating my parental genets, I'm replicating check genets. So it would take a lot of area to replicate my parents multiple times. So instead, we just replicate these checks in each incomplete block. And those checks are used to adjust for spatial variation. So we get better estimates for the values of each parent. This experiment is planted at 2 locations. One is in Lamberton, Minnesota, which is the southwest part of the state, and the other one is on St. Paul. If you're driving by the St. Paul campus, it's on the corner of Cleveland and Larpenteur, if you're curious to see what it looks like. So what have we done so far? Last year, we cloned and planted. So in May, we cloned from parent genets. Then in May through July, we maintained them. In August, we transferred them to peat pots for transplanting. And finally, in August and September of last year, we actually planted this experiment. So this year is the year where all the fun things start. We get to finally phenotype, and we're planning on phenotyping for 2 years. We'll look at things like vigor, heading, anthesis, or the date of flowering, plant height, it's also an important one, grain yield, and how it persists over multiple years. We'll probably do that on some sort of like 10 spike average and maybe a whole plant average also. Lodging, we don't want our plant to be laying over in the field. Spike traits, so the length and the density of the spikelets or of the florets on the spike. Seed traits like length, width, and weight. We'll use the Marvin optical seed analyzer to do a lot of that. And the final 2, which are shatter resistance and threshability. And these 2 I'll delve a little into with a little bit more detail. Threshability. So previously we've scored threshability using a 0 through 9 scale, 0 being 0% free threshing, 9 being 90% free threshing. So up here on the right, this would be something that would not score very well. You can see that most of the seeds still have their holes, whereas here, a lot of the seeds are D-holes. So this would get a better score for free threshing. The Younger's lab on campus actually has a pretty slick image analysis pipeline that we're hoping to try out with a digital camera and an R script that gives you a percentage hold D-hold seeds. So we'll probably try that out this year to see if we can get a better estimate for our percentage hold or D-hold seeds. Shattering is also another really important trait. And it's important to differentiate the fact that there's multiple types of shattering. So I know Praveen in our breeding program has just given genets a general rating 0 through 9 with how well or how resistant they are to shattering. But I think it's going to be important to separate the types of shattering, which Kayla Altendorf characterized these in her thesis work last year. But floret shattering and brittle rachis are 2 types. Floret shattering is under more complex genetic control, so several QTL are controlling floret shattering, whereas brittle rachis is under simpler genetic control. So I'll probably give each of these types of shattering a rating. So I'll have ratings for floret shattering and ratings for brittle rachis shattering. And lastly, what are we going to do with all of this phenotypic data? We're going to use it to assess breeding progress. This breeder's equation that I have here is the most boiled down simple version. But basically, all we're doing is predicting genetic gain or a predicted response to selection, which is R. And that's equal to heritability, which heritability is just the proportionate variants that are due to additive genetic factors, and S, which is our selection differential. So it's that mean of the selected individuals minus the mean of that cycle that we're selecting from. And this gives us a prediction or predicted genetic gain. And we can use this to look at how much genetic gain we've had between each cycle and also predict genetic gain for future cycles of selection and maybe change how we're doing things a little bit based on what the results of this are. So I've talked about assessing breeding progress. Now I'm going to continue the breeding talk and talk about breeding or genome-wide association study and comparative genomics to identify regions of the genome that are associated with important domestication genes. So genomic resources. I know this is the intermediate wheatgrass is a new perennial grain crop, but we do have genetic resources. Intermediate wheatgrass has a large 12 gigabase pair genome. It's an allohexaploid, so that presents a number of challenges just based on those two things. But we have our version 2 of the annotated is an annotated genome. The details are here on the right. And there's a new genome that's in the pipeline. It's almost ready to publish that uses PacBio long reads with really great coverage, 34.9x coverage. So we have a good genome in the pipeline that we'll get to use. And we do have genomic resources, even though it's a relatively new crop. So there is a lot of work that's been done characterizing some domestication traits in intermediate wheatgrass. The two papers I have here from Steve Larson, genome mapping of QTL controlling domestication traits, and also the thesis work of Kayla Altendorf with her NAM population. However, there are many candidate domestication genes in other species that align well with intermediate wheatgrass QTLs. For example, free threshing, we found alignment between barley, wheat, and rice genes. You can see that here, looking at free threshing. There's a number of genes there, along with brittle rachis, the barley, brittle rachis 1 and 2 genes too. So there's many candidate domestication genes in other species that we've seen align with intermediate wheatgrass QTLs. So we could apply this information to our own breeding program to see what we've been selecting for and fixing in our own population over time. Our method of genotyping is called genotyping by sequencing, or GBS. GBS is just a reduced representation method of sequencing. It's really cost effective. We've used it with great success in our program. And the output from GBS is a SNP file. For those who are not breeders, a SNP is a single nucleotide polymorphism, or basically just a difference. It's a list of all the differences in an individual's genome, how it differs from the next genet, right? So a SNP is a polymorphism or a difference. And so a genome-wide association study basically takes those differences or those SNPs and associates them to a trait of interest. For example, we could pretend that this Manhattan plot, which is a typical output from a GWAS, we could pretend that we're looking at shattering. And so what this would tell us is that at chromosomes 19, 12, 8, 6, and 5, it looks like there are some significant SNPs that are significantly associated with our phenotype of interest, which is shattering. So then we can delve more into what each of those SNPs is and potentially identify some QTL for those traits of interest, which in our case is shattering and threshability. Which leads me into comparative genomics. Comparative genomics is identifying DNA sequences that have been conserved in many different organisms over millions of years. So we could compare domesticated species to undomesticated species and compare shattering and threshability across species, which is obviously a really interesting application for our research. So we could compare the intermediate wheatgrass genome to the genomes of wheat, barley, rice, brachypodium, discover syngenous regions with known domestication genes, look at our allele frequency changes between each cycle and other signatures of selection. And finally, how do we actually use this information? This information can be applied to accelerate the breeding and domestication process of intermediate wheatgrass. So we can directly target genes in intermediate wheatgrass that are homologous to domestication genes of other species, wheat, for example, and those arose randomly and were fixed. But we can use our 21st century tools to accelerate the process of breeding of intermediate wheatgrass. So we may be late to the game, but we have a lot more tools in our tool belt that we can apply to speed up the domestication of this crop. All right, I'm going to switch gears a little bit and talk about pre-harvest sprouting, which is an important agronomic trait in intermediate wheatgrass. So pre-harvest sprouting is basically just the premature germination of a seed in the field. For obvious reasons, if a farmer is going through their field combining, they don't want to see that. They would get docked heavily for grain that's already sprouted prior to harvest. So it's a non-desirable trait, and it's been really well characterized in wheat and barley, and we actually screen for it in our breeding program for wheat here at the University of Minnesota. And we screen for it early, and those lines which have a propensity to pre-harvest sprout are eliminated from the breeding program, so we don't end up with cultivars that look like this here on the left. The protocol that we use for pre-harvest sprouting basically involves harvesting at physiological maturity, drying it down, then putting it in the dew chamber for around a week, and then we rate it for pre-harvest sprouting. 0 being no PHS, 9 being about 90% sprouted grain. So we have a really established protocol for wheat. However, it's has not been characterized in intermediate wheatgrass basically at all. So that's why this research is important. PHS in intermediate wheatgrass, we know that we've seen it before. You can see in this picture here, some roots coming out. But intermediate wheatgrass has a number of challenges that I think we'll have to figure out as we figure out the best way to screen for PHS. And the first is harvest methods. So there's multiple ways you can harvest intermediate wheatgrass. This research is ongoing. The Younger's lab on campus, for example, is looking into what might be the best recommended way to harvest. One way would be to combine directly, which would be here on the bottom left, how you harvest beans or corn. Another way would be to swath or to cut and then put it into windrows and then combine. So this combine here is combining a windrow. And things can sit in the windrows for several days, if not weeks, before being combined, which allows it to dry down, or in some cases, begin to pre-harvest sprout if the humidity and rain or whatever, anything like that is right, things will start to germinate. So we need to figure out the best way to screen for PHS in intermediate wheatgrass so we don't have farmers that end up with grain that looks like this. Right now, we're planning on using our protocol that we have for wheat in 2021 to try to screen for materials. And we might also try a pilot study on a few intermediate wheatgrass genets, sampling at different maturities or different days after physiological maturity to see what might be whenever we see the most pre-harvest sprouting. But some considerations and challenges of pre-harvest sprouting in intermediate wheatgrass are that the maturity of intermediate wheatgrass is really variable. So within the plot, there's a lot of variability of maturity. And even within the spike, there's a lot of variability for maturity. So you might have some florets that are flowering and some that are setting seed within the same spike. So that offers a number of challenges for deciding when the optimal time to harvest and when physiological maturity actually is. But nonetheless, it's still important. It's would be really useful if we could figure out a way to screen for this in our breeding program. And if not that, at least make some recommendations for farmers as to how long they can leave their Kernza and windrows prior to harvesting, or best avoid PHS in their grain. Okay, the last part, developing Kernza education and outreach modules. I'm going to wake you all up with another word cloud that I'd like to see you all build. And I'd like to know how much you've retained from my talk. So what does it take to commercialize a new crop? You can join the poll via text or web browser. And the same goes for this one. Communication, I like that. Markets, customers, love. I see markets is growing. Agronomic knowledge, cultivars, interest. Yeah, that could be consumer interest. Communication, market demand, integration, supply, quality, markets, education. I see education getting larger and larger. All right. A few more seconds. Genetics. Awesome. Thanks, everyone. You've retained a lot. Yeah, we have to advance germplasm and trait evaluation, agronomics, environmental, education, extension policy, supply chain, markets. You got it all. It's a team effort commercializing a crop. So this final portion that I'm talking about is engaging education, extension, and policy, another important part of the successful commercialization of a new perennial grain crop. So I've been working with Aubrey Straight-Krug and Megan Gladbach at the Land Institute. This figure was created by them, so they get the credit for this awesome visual for how they plan on rolling out these educational modules to basically show people how Kernza fits into our community, our agroecosystem, and our earth systems as a whole. So right now there's a number of different modules that are going to be made. And the two that I will likely be making are perennial grains from farm to plate, and the appropriate one for my research, which is Kernza breeding and genetics, which is obviously right up my alley. These will be some sort of online module format, and we'll probably release them to a smaller number of people, do some beta testing, reassess, change things around, make sure all of our learning objectives are being met, and then re-release to the public so people can learn about how Kernza fits into these three things. In addition to educational modules, I've also been working with Connie Carlson with grower researcher networking. So making sure that our growers and our researchers are sharing information both directions is really important, especially as we look forward to the commercialization of this new crop. So we need to make sure that those learning needs are met and well communicated. That's been kind of what I'm planning on doing, what I've done previously, surveying people to figure out what the needs are and make sure that they're being met via field days, other meetings, things like that. So education is really important for the success of a new crop, and I'm excited to be a part of a really big, really awesome team who are working towards this goal. All right, in summary, I gave you all an introduction to intermediate wheatgrass. Then I talked about breeding progress and how we can predict genetic gain for future cycles. Then I talked about GWAS and comparative genomics for domestication genes, and how we can use this information to speed up the domestication process. Then I switched gears and talked about pre-harvest sprouting and how we might be able to use this to screen our breeding materials for PHS, and maybe glean some information that's useful for growers. And lastly, I talked about developing Kernza education and outreach materials and how important this is to strengthening the intermediate wheatgrass knowledge network and to educate the public as a whole on perennial grains. Thank you everyone for coming today and sitting through my talk. I'd like to thank my sponsors, FFAR, which was a major grant for this research, the Forever Green Initiative, Minnesota Department of Ag, University of Minnesota, and the Kernza CAP grant. My advisor, Jim Anderson, my committee members, Jenny Kimball, Jake Youngers, Jess Gupnecht, and Praveen Bajgain. And importantly, the intermediate wheatgrass team. Brett Heim is an awesome technician. He's kept everything in great working order in our intermediate wheatgrass breeding program here at the U. Praveen, who is actually the breeder who's doing a lot of these selections and improvements. He's the one actually doing the genomic selection that I talked about earlier, and making really great gains for the program. And a lot of undergraduate workers who have helped with anything and everything you can imagine. And lastly, the Anderson Lab group, our technicians, Susan, Nate, Brett, and Emily, postdocs, Praveen, Anthony, and Ratan, graduate students, Emmanuel, Zenith, Max, and Matt, and again, numerous undergraduate students who have helped make these dreams become a reality. And thank you all for attending this talk today. I'm glad you're here. And I'd love to take any questions that you might have and answer them to the best of my ability.
All right. Thank you, Hannah. That was a very informative talk. If you guys have any questions. So there's a question. What exactly are your replicated checks?
Awesome. Yeah, what exactly are your replicated checks? That's a great question. So these are actually just randomly selected. A couple of them are border plants, so they aren't the same as they aren't the same identity as any of our plants. But off the top of my head, I couldn't tell you anything special about any of those checks. Just know that they were clonally propagated, so each check is technically a clone of one another. If that wasn't a good enough answer, feel free to continue messaging me and I'll try to provide more information.
All right, well, we're waiting for questions. So I'll shoot in one question for you, Hannah. In slide 16, you have a figure that, there's a breeding improvement in shattering trait. So I know usually shattering trait, there are changes in the abscission layer. So do you know if there are any changes in the abscission layer?
Yeah, I think that's probably what's controlling a lot of that shattering is the formation of abscission layers in different parts of the plant. I don't know that anyone's specifically characterized the abscission layer in intermediate wheatgrass. Someone can correct me if I'm wrong, but yeah, that would be my assumption, that it's largely controlled by that.
So another question here. Is it likely that future breeding will focus on grain quality in terms of increasing gluten content so that the flour can be used in a wider variety of application, or could realistically replace less environmentally friendly wheat?
Yeah, that's a great question. And I'm sure there's probably some people on the commercialization team on this call that might be able to answer this better than I could, but I don't know that the goal is to completely replace wheat per se. And I don't know that we'll ever get to the same gluten content as wheat. We'll make improvements for sure, but I don't know that we'll ever get there. And there are a lot of other uses for Kernza. Rather than making your bread rise, you can use it in Kernza or in crackers, cereals. I personally used it in banana bread, and it makes a really bright banana bread. So I guess I don't know if that's the ultimate end goal. My hunch is no, but.
Okay, so there's one more question. What are the implications for plant breeding? So I think
I don't know if whoever asked that could clarify the question, I guess.
Yeah, it's anonymous, so we'll wait.
Specific implications if somebody wants to maybe amend their question.
So there's one question from the chat from Kayla. What population you will be using for GWAS?
Awesome, that's a great question. So the population, I apologize if I didn't clarify that enough. I'll be using the parent population. So parents from cycle 2, 3, 4, and 5, which I know will offer a number of challenges using multiple cycles for a GWAS, but that's the plan.
Okay, another question from Dom. Is there any interest or importance in phenotyping rhizome and tiller density or differences in rooting depth / biomass?
Nice. Interest in phenotyping rhizome and tiller density. We do take notes on that. We don't want to see something that's tillering out too much uncontrolled. And then differences in rooting depth biomass. I would say we have not from the breeding standpoint, no, we have not been like breeding for something with more or less root biomass or root depth. But it's kind of the reality of the plant as it is now.
So there's one question from Grant Chansons. How do you plan on increasing second year and third year yields?
That's a really great question. I think a lot of other breeders on this call, we're all kind of working towards that same end. I don't know that I have a specific, this is how we're going to do it. This is how we're going to increase yields. But again, that's the big challenge of this crop. We're reallocating photosynthate from the roots to the grain, and kind of going against the nature of what a perennial wants to do, wants to put that energy down into the roots rather than the grain. So I'm sorry, I don't have a better answer to your question yet, other than our breeding efforts are trying to sustain that yield for the second and third year.
Okay, so if you have more questions, you can shoot it in the Q&A box. And while waiting for another question, I want to ask one more question for Hannah. So you said you're interested in doing the education and extension part of like the commercialization. So do you have any field days for Kernza or like demo plots so the farmers, like I haven't, or for example, like farmers that are not familiar with the plant or the crop? Yeah,
there should be some field days this summer. I don't know that the dates are set in stone, but yeah, that's a big part of it, is getting people exposed via field days. In addition to like, we had grower calls in February of this year, which were really productive for growers and researchers to ask questions, share knowledge, things like that.
Thanks.
Could you tell a little bit about the work that Colin Curtin is doing and working with farmers to go through that process of selecting farmers that are going to grow? Could you say a little bit about that?
Yeah, so Colin, I don't know if I'm answering exactly what you're saying, Don, but so Colin Curtin is working on the commercialization team along with Connie Carlson, to choose growers and target where these where we're growing Kernza. Can you clarify, Don, if I'm not answering?
Well, I was just talking about, field days. And so that's the group is, developing the field days along with the rest of the team. And that's really where the educational process is occurring, right? And Colin's work with the farmers as he goes ahead and provides information to them about where to plant, how to plant, and all of those things, right?
Yeah.
So we have one more question from Kayla. How many individuals are in your parent nursery, and what inspired you to do the augmented design?
That's a great question. I think the number was 266, so under 300 in the parent nursery. And what inspired me to do the augmented design, so an augmented design saves field space. It's less work to replicate each parent because Kayla, I know you know this, but you actually had to split each parent to replicate at 2 locations. So that would have required another split to, for example, do a randomized complete block design or something like that. So an augmented design helps us or allows us to save some field space and some effort separating out parents. It's not like wheat where we can just make a few more envelopes of seed to have an additional replicate.
All right, so one more question from Grant Johnson. So are there any important pests and diseases associated with Kernza?
Yeah, so as far as diseases, you occasionally see FHB, ergot is more common on the edges of your field where things are not pollinated as well. So I used to work in hybrid wheat and saw ergot a lot, and it's whenever something isn't fertilized with pollen, you see ergot more often. Pest diseases. So so yes, there are some diseases, but in general, it's relatively disease resistant.
So one more question. Is it possible that the harshness of Minnesota winters plays into yield reduction in subsequent years, or Is that something that breeders are considering?
I don't think that it would be the harshness of Minnesota winters that's reducing that yield. I think it's more so, which I guess this to answer someone's previous question, I think that the yield reduction, it's not just a breeding question, it's also an agronomic question, and figuring out what the best way, what the best distance between how we're planting is, things like that. Because as this plant grows, it creates more, it spreads across the ground, right? And I think that that's influencing how we're shifting from increased grain yield to more resources and underneath the soil. So I think it's kind of an agronomic and a breeding combined effort to get those yields to persist. But I don't think that it's necessarily a harshness of Minnesota winter that's causing that.
Okay. Do you think it would be beneficial for nutrition security by solving nutrient deficiencies?
Nice. Thanks for your question, Batul. I know Batul has some great experience learning about zinc in wheat also. I don't know. I guess, do you think it'd be beneficial for nutrition by solving? I don't know that it would solve any major nutrient deficiencies that anyone has. If there's any food scientists on the call and you want to chime in or share your knowledge, it's guaranteed to be more than mine is on this. I don't know that it would solve any nutrient deficiencies, but it is a relatively nutritious grain, especially consumed as a whole grain.
So another question from Kayla. What method will you use to evaluate flowering time?
Yeah, that's a really great question. So I haven't collected any data yet. All of my experience collecting data on flowering time has been on red wheat, which flowers right around the same time, which is a challenge for intermediate wheatgrasses, that things flower at very different times. So my thought right now is that I might do some sort of rating and like Julian, so days after January 1. Maybe I'll do like 25% of the plot is flowering at this date, and then 50% at this date, 75% at this date, something like that. But I don't know that I have my methods completely nailed down for that.
So here's the question from Rex. If yields are poor in year 2, is it economically feasible to simply treat intermediate wheatgrass as biennial crop or year 3?
Yeah, it maybe that would be ideal. If Colin Curitin's on the call, he knows a lot more about the economic viability and what the trade-offs would need to be for that. But I think that answer will probably change as we release more intermediate wheatgrass cultivars. There's already a lot that are a lot in the pipeline that are looking a lot better than in Clearwater, and maybe as we see higher grain yields, that might change some of those economic trade-offs. But that's a good consideration. Maybe the end goal is going to end up being a 2-year crop rather than trying to have 3 years.
So I don't see any questions in the chat and in the Q&A box. Great job, Hannah. You did very well. So last minute, going once, going.
These have all been really great questions. Thank you, everyone.
Yeah.
And I apologize if I didn't answer them the way you wanted to, but feel free to follow up with me via email. I'd love to keep chatting.
Oh, there's one more. What would you rotate Kernza with?
Yeah, so there's as far as cropping rotations, there's some research on legume intercropping. Jake Youngers could probably talk more on that. But I don't know what your trade-off would be after those 3 years. I think that research is still ongoing, but also
I think one idea that's being developed is to develop a continuous perennial rotation with alfalfa. So as a lot of you know, alfalfa can provide the nitrogen for about 2.5, 3 years of corn. So they're thinking of potentially incorporating that model in rotation with Kernza. So it'd be potentially an 8-year rotation or a 7-year rotation with alfalfa and Kernza. So there's a lot of, Jake is probably on today, but Jake is doing a lot of work in those types of rotations.
Yeah. We're learning as we go. It's really awesome. I think Jim had his hand up earlier. I don't know if he still does.
So I think that's all. Great job, Hannah, and thanks everyone for tuning in. Great discussion, great questions.
Yeah, thanks everyone.